Welcome to TheBioInsights, where science comes alive.
In a typical clinical or environmental sample, millions of different bacteria are mixed together in a dense, chaotic soup. To successfully identify or treat a specific pathogen, you have to extract exactly one microbial cell from that mess and grow it by itself. This image shows what success looks like. On one side, you have a heavy, blended smear of growth, but on the exact opposite side, you can see perfectly circular, distinct dots. We call these isolated colony-forming units, or CFUs. Each one is a pure cluster of millions of identical clones, grown from one original cell. Before you can isolate a target, you need a clean, disinfected workspace. Put on a lab coat and gloves, tie back any loose clothing near your Bunsen burner, and gather your tools. Use a labeling pen to mark your sample details directly on the bottom of the Petri dish, never on the lid. Lids can easily get dropped or accidentally swapped to another plate. You also need to let your nutrient agar plate reach room temperature prior to inoculation. Exposing fragile microbial cells directly to a cold gel matrix will temperature shock them, drastically lowering their survival rate. If you simply rub a sample swab randomly across that agar, everything will grow over each other into a useless mass. To physically separate one cell from the noise, we have to use a precise mathematical dilution technique called the four-quadrant streak. To start, we need to achieve total sterilization of our primary tool, the nichrome wire loop. Hold the metal loop directly inside the open flame of a Bunsen burner until the wire glows intensely bright red. Once it is sterilized, pull the loop away and let it cool in the ambient air. Do not blow on it to speed up the process. If you touch a hot wire to your sample, the heat will instantly boil and kill the exact cells you are trying to recover. To safely verify the temperature, gently touch the hot loop to a clean, uninoculated edge of the agar inside the Petri dish. If the gel sizzles, wait a few more seconds. Once cooled, collect a small amount of your bacterial sample. Hold the metal loop lightly in your hand, exactly like a pencil, keeping the shaft tilted at a 45-degree angle to the plate. The nutrient agar has the physical consistency of firm gelatin. Glide the loop over the surface smoothly. Applying too much downward pressure will easily cut or tear the gel matrix. This diagram shows the exact motion for your first pass. Start at the top edge of the plate and drag the loop back and forth in a tight, dense zigzag pattern, covering exactly 1/4 of the plate's total surface area. This initial smear forms quadrant 1. It establishes a highly concentrated reservoir. Every single cell we spread across the rest of the plate over the next 3 steps will originate from this dense patch. Place the lid back on your plate to protect it from airborne contaminants. Now, place your loop back into the flame until it glows red, let it cool completely, and test it again on a blank spot of agar. We need a completely sterile tool so no new bacteria enter the gradient. Pick up your Petri dish and rotate it exactly 90 degrees. This moves the heavily saturated quadrant 1 to the side and places an area of fresh, sterile agar right in front of your hand. Looking at our blueprint again, place your sterile loop on the outer edge of quadrant 1. Drag the loop through the original streak lines exactly 2 to 3 times to pick up a tiny fraction of the bacteria. Then, streak a new, slightly looser zigzag pattern across the adjacent empty quarter. This dragging motion strips away a fraction of the cells from the first quadrant and spreads them out across a new, wider surface area, significantly reducing the density of the bacterial population. If your lab uses disposable plastic loops instead of metal ones, you skip the Bunsen burner. Simply throw away the used plastic stick after quadrant 1, grab a fresh, pre-sterilized loop from the package, and proceed straight to quadrant 2. With either tool, we repeat the procedure. Flame and cool the metal loop, rotate the dish another 90 degrees, overlap the edge of quadrant 2 exactly 2 or 3 times, and streak a third zigzag pattern into the next empty quarter of the plate. Through each successive rotation and pass, the geometric reality of this method works in your favor. You are intentionally leaving the vast majority of the cells behind, carrying a smaller and smaller fraction forward into each new sector. Now we set up for the final pass. Sterilize your loop in the flame, let it air dry and cool off, and give the plate one final 90-degree turn. For this fourth quadrant, the rules change. Your sterile loop must cross the edge of quadrant 3 only one single time. As the diagram shows, you touch the very edge of quadrant 3 just once, and then drag the loop in a wide, sweeping zigzag pattern. Use up all the remaining empty space in the center of the plate, and space your lines out loosely. You must watch your loop carefully during this final sweep. If you bump into the heavy growth of quadrant 1, you'll drag millions of cells back into the low concentration zone and ruin the dilution. By crossing the line just once and spreading the streak out wide, you guarantee that the final surviving cells clinging to your loop are deposited far enough away from each other to grow into perfectly isolated structures. With the physical streaking complete, immediately securely place the lid back onto your Petri dish to cut off any exposure to dust and airborne contaminants. Before it goes into the incubator, there is a strict orientation rule. You must flip the Petri dish completely upside down, so the agar sits flat on top and the lid rests on the bottom shelf. The plates stay in the incubator at 35 to 37 degrees Celsius for 24 to 48 hours to allow the individual cells time to multiply. This diagram shows exactly why we invert the plates. If you incubate a dish right side up, the heat causes water trapped inside the agar to evaporate and condense on the cooler plastic lid. Those water droplets become heavy and drop straight back down onto the flat agar surface below. That falling water washes across the surface of your plate, picking up your separated cells and merging them back into a blended, useless soup. Keeping the plate upside down traps the condensation safely in the lid. After 24 to 48 hours, remove your plate and inspect the surface. This comparison shows our goal. Looking at a properly streaked plate, you will immediately see a heavy, solid wall of bacterial growth packed tightly into your first quadrant. Follow your streak lines visually across the agar. You can watch that heavy wall thin out across quadrant 2, break apart even further in quadrant 3, until it finally resolves into clear individual dots in the final sector. Because those final dots are sitting separated from one another, surrounded by empty agar, each one represents a pure culture that multiplied from a single original cell. By meticulously diluting that messy starting sample down to a solitary unit, you have established a pure culture. You are now fully prepared to extract a pure sample and proceed to gram staining and identification.